Hi, everyone. Thank you for joining me for this talk on microbiome manipulation in the type 2 diabetic patient population. I am Dr. Michelle Maddox, the clinical consultant for Omni-Biotic, and it's a pleasure to be with you today. Today, we're going to do a brief review of terminology to make sure that we're all on the same page and using the same words. We're going to spend a few minutes reviewing the scope of the problem of type 2 diabetes. We will review the interactions of the gut with other body systems, and we'll explore the state of the current literature as it relates to type 2 diabetes, comorbidities, and the gut microbiome. And finally, we'll spend just a couple of minutes discussing how to choose a good high-quality probiotic and how to explain why your patient should be choosing one product over another. So again, in the interest of clarity, let's review a few basic definitions so we're sure to all be on the same page. The first picture here is the microbiota, and literally this is just the organisms. The next picture is showing you the metagenome. These are the genes and the genomes of the population. So that's both the bug and the host. And again, the genome is just the sum total of the organism's DNA. The last picture is the microbiome. This is the gene, the genomes, and the products of the population. Again, both the bug and the host. This is where we are looking at that whole soup, not just an individual ingredient. And this is looking beyond the bugs into what they're actually doing, that metabolomics. You'll hear me say the word strain a lot in this particular presentation, so let's take a moment to discuss what I mean by that. For some of you, this will be a review, but for a lot of you, this is going to take us back to basic biology. I know when I took biology, we didn't talk about strains. So most of us learned some version of King's Play Chess on fine-grained sand. That's the kingdom, phyla, class, family, genus, and species. You'll notice that there is no strain mentioned in this taxonomy. A strain is isolated from a species. It's distinguished by genotypic and/or phenotypic characteristics. Specific strains are then tested for very targeted outcomes. Let's take a minute to look at the types of diabetes and review just some basics about them. Type 1 diabetes involves both innate and adaptive immunity, and it's targeting the pancreatic beta cells. The triggers can be genetics, the environment, microbiome, immune, and metabolism. Type 1.5, which is the latent autoimmune version, it's a bit of a hybrid, and it includes both type 1 and type 2 characteristics. It's most often seen in obese adolescents with type 2 diabetes, and they also have positive antibodies. And those antibodies are usually only seen in type 1, and so that's what makes this a bit of a hybrid. So type 2, this is often referred to as acquired. There are no antibodies present. And again, triggers can be obesity, genetics, the environment, microbiome, the immune system, metabolism, etc. Gestational diabetes happens to moms even with normal BMIs and no history of diabetes or prediabetes. And in gestational diabetes, there are higher rates of C-sections, stillbirths, developmental delays, obesity, diabetes for the child, as well as higher rates of diabetes and negative cardiovascular outcomes for the mom. In type 3, this is diabetes secondary to either a disease or an injury to the pancreas. I have a dear friend who had a really nasty mountain bike accident where she actually lost 3/4 of her pancreas. And as a result, despite the fact that she is very active and eats incredibly well, she has now been pre-diabetes and every now and then has to really struggle to stay in the pre-diabetes and not fall into diabetes because she only has a very small amount of her pancreas left. Here are a good couple of diagrams on the different types of diabetes. I am highly visual, So when I see good diagrams, I tend to pop them into presentations. I know I'm not the only visual person out there. So let's talk a little bit about the scope of the problem. Most of us are aware that diabetes, especially type 2 diabetes, is a huge issue in the world. Let's spend a few minutes looking at the numbers and at the specifics. It's really quite staggering. In 1980, there were 108 million people with type 2 diabetes. In 2017, there were 451 million people with type 2 diabetes. By 2017, diabetes was accountable for 12.8% of all-cause mortality in the world. And gestational diabetes impacts up to 14% of all pregnancies. And again, with gestational diabetes, there's a significantly higher likelihood both the mother and her offspring will develop type 2 diabetes within their lives. Of the people who have diabetes, 90 to 95% of them have type 2 diabetes. The map in the upper left is showing the distribution of type 2 diabetes across the globe in 2017. The charts on the bottom right, the first chart is showing the incidence, which are new cases, of type 2 diabetes by age. That's the orange line. And they're comparing it in that same chart to the prevalence, which are the new and the existing cases in the gray bars. The chart next to it, the dotted line shows the incidence, so again, new onset cases, by age in 1990. And the orange line is showing that same statistic, the incidence by age, in 2017. It is really quite clear to see that the incidence and the onset of type 2 diabetes is trending younger and younger in lives. This map shows the number of people with type 2 diabetes across the world in 2013 and the projected numbers for 2035. This is based off of the data in 2013. I could not find one for 2017 or even closer than 2013. My strong suspicion is that these numbers have gone up probably fairly drastically. This is looking at comorbidities of type 2 diabetes. Obviously, this chart is not exhaustive. There are a very clear top 4: hypertension, back pain, retinopathy, and depression. The remainder of the comorbidities are clustered down at the bottom of this chart, and they are in the less than 15% of people mark. This chart is also looking at comorbidities, but it's looking at comorbidities over time. Not surprisingly, we see the number of comorbidities rise the longer a person has type 2 diabetes. This is another really great visual, and it's a diagram of the impact diabetes and hyperglycemia can have on the brain and our systems. Hyperglycemia creates oxidative stress, which can trigger depression, and this is via the monoaminergic system. This system is involved in behavioral arousal, attention, motivation, control of motor, and autonomic processing. It triggers anxiety via the GABAergic system. This system's involved in regulation of vigilance, anxiety, muscle tension, memory function. It regulates cognitive and emotional processing. And then hyperglycemia also triggers memory impairment via the cholinergic systems. And these are involved in emotion, cognition, locomotion, sleep, attention, arousal, and neural activity via those acetylcholine receptors. In all of these, you see that the antioxidant defense system is triggered by each of the aspects, and you'll see the corresponding oxidative stress products like that superoxide dismutase. So there are a number of difficulties in managing type 2 diabetic patients. It's hard for the patient when they get diagnosed, and it's also incredibly difficult for the doctor to manage them. There's a really wonderful article that interviewed patients with type 2 diabetes to assess how they were managing and what their thoughts and their feelings and kind of what they were going through. I very strongly encourage you to pull that article. It's referenced on this slide. On the left side of this slide are some quotes pulled directly from the article from the patients with diabetes. In general, patients are embarrassed. They feel like there's a negative social stigma with type 2 diabetes, like they're a failure at life, or they feel very guilty that they have it. They feel very overwhelmed. Treatments and the changes are massive, like changes to diet, medications, exercise, lifestyle, daily foot checks, etc. It's even more challenging if the family is not supportive. They now have dietary restrictions that others either resent or that the rest of the family doesn't do. So now they feel very deprived. There's absolutely the issue of healthy food is expensive, right? It is significantly more expensive in our country to eat healthy than it is to eat really low quality food. And then you definitely have this aspect of not having time. They don't have the time to cook or to learn how to cook. They don't have the time to exercise. They don't have the time to check my diabetic mother's feet, was one of the quotes. And then for a lot of them, they feel this sense of hopelessness. They expressed in these articles a lot of, everyone in my family has it, and therefore there's no way for me to avoid getting it. One of my loved ones died from diabetes, so I will too. It's a life sentence. The changes are too hard to keep up for my entire life. If you have a moment, I really encourage you to look at these articles. It gives another version and another insight into what your patients are struggling with so that you can meet them there. It's not any easier for practitioners who are managing type 2 diabetic patients. And I'm betting a ton of you do this. And how many of you actually have patients who are compliant on a long-term basis? Hopefully, when you realize that they're not being compliant, you are asking them why in an open and non-judgmental way. And you're also asking yourself why to see where you can improve. The things that we tend to see is that practitioners don't know how, or they don't have the time to give very specific diet and exercise recommendations. They don't feel qualified because of how their practices run. They don't have the time to give that information. A friend of mine, even though she has the knowledge, she doesn't have the time. And therefore, she's partnered with this amazing nutritionist who she refers her patients to. They share patients, they work really well together, and her compliance has gone through the roof. Build your network of referrals. If this is you, if you feel like you don't have the expertise or the time, build that network of referrals, people you can refer back and forth to that have the knowledge and that will take the time. The current diagnosis and treatments neglect a huge part of the picture. They don't look at inflammation or oxidative stress. They do not address eating disorders. They do not address the psycho and emotional issues. Remember, depression was in the top 4. There's also a large degree of who's on first. So with this disease, patients tend to see a large number of practitioners, and it can become confusing for the practitioners as well as for the patient who's in charge of what's happening for them. So is it an endocrinologist? Is it their internist? Is it the family doc? Is it their podiatrist? It's a little bit difficult when they're juggling so many different people. And finally, what happens when you haven't seen this patient for 3 months, 6 months, 12 months? Does your system alert you to reach out to them? Or do you assume that they're being managed by somebody else? Gut microbiome alterations seen in type 2 diabetes. Let's focus on that. This is a microbiome talk after all. So overall gut microbiome diversity goes down. The makeup of how many different types of bugs in the gut goes down. I will take a moment here to discuss the concept of commensals versus pathogenic bacteria. Commensals, I always think of them as who's in your community, right? Commensals are organisms that, frankly, they're supposed to be there, but they're supposed to be there in a balanced way. Most commensals have the ability to turn into bad seeds or potentially pathogenic organisms if they are too high and they get out of control. So we can see organisms like Akkermansia, Faecalibacterium, and obviously most of us have heard of this Firmicutes Bacteroides ratio, especially when it comes to obesity and gut microbiome alterations. We see that the microbiome, which controls short-chain fatty acids, these balance of short-chain fatty acids become skewed. The main short-chain fatty acids we all tend to focus on are acetate, propionate, and butyrate. And we often see butyrate, which is the preferred fuel of colonocytes, and it's super important for barrier integrity, go down. We see things like lipopolysaccharide or LPS translocation. This is a product, it's an endotoxin of bacteria, and it's able to pass through the gut because there's now an impaired permeability. J&K is an inflammatory kinase, and it's activated by glucose, cytokines, and free fatty acids. It's crucial to mount a proper response to stress. You need this. However, elevations have been associated with obesity and obesity-induced insulin resistance. IKK complex, it's activated by inflammation, so TNF-alpha, interleukin-1, microbial products, and again, stress. It in turn activates NF-kappa beta. This complex has been associated with obesity, metabolic syndrome, type 2 diabetes, cardiovascular disease, and NASH and non-alcoholic fatty liver disease. I jokingly refer to this next section as how many gut axes are there? And it's true, there are so many interactions between the gut and other systems and organs, and we are really just beginning to wrap our heads around it. Let's spend just a brief amount of time reviewing a few of those that are particularly relevant to this patient population. So this is an application that we have all known about for years. It's responsible for digestion and metabolism, the degradation of insoluble dietary fiber, protein, degradation, fermentation, bile acid deconjugation. It helps us synthesize vitamins, can help regulate appetite and bowel regularity. It's also counted on to activate certain medications. This is why people are always told when they're taking oral birth control that you need to use another form of birth control when you're on antibiotics. The oral birth control pills are formulated so that they are actually activated by gut microbiome and those microbes in the gut. And if you kill the microbes in the gut, that oral birth control pill may not activate properly. Again, highly visual. This particular slide, I think, is a really good slide. And frankly, it's really helpful for patients. And you can see in this high fat, highly processed, pretty standard American diet on the left-hand side, it leads to things like LPS and cytokines and inflammation. The high meat intake without a good balance, right? That can lead to things like TMAO and increased risk of cardiovascular diseases. And finally, the one on the end, which is a high fiber, predominantly plant-based diet, that leads to healthy balances of those short-chain fatty acids, higher amounts of those good commensals, and anti-inflammation and anti-anxiety products. The gut-liver axis, this is huge, and especially again in our type 2 diabetics, right? Primary bile acids are conjugated to secondary bile acids by bacteria in the gut. Receptor FXR and TGR5 are activated by bile acids. These receptors are responsible for glucose and lipid metabolism. Intestinal permeability is up and down regulated by bile acid and AMPs. AMPs are antimicrobial peptides, acetaldehyde, LPS, TMA, etc. Increased intestinal permeability allows translocation of things not supposed to leave the intestine. Remember, we talked about translocation of LPS. Things like PAMPs, which are pathogen-associated molecular patterns, and DAMPs, damage-associated molecular patterns. They're collectively known as MAMPs. There's a lot of little acronyms here. These molecular patterns are recognized by the Kupffer and hepatic stellate cells. They trigger inflammation and have been very strongly linked to liver fibrosis. Xenobiotics and steroid hormones are conjugated and packaged for excretion in feces. Bacteria in the gut produce an enzyme called beta-glucuronidase. This enzyme deconjugates them, freeing them up for recirculation. And then finally, short-chain fatty acids are friends. So again, these are things like butyrate, acetate, and propionate. They're produced by gut microbes acting on resistant starch, i.e. fiber. These short-chain fatty acids, especially butyrate, are key for maintaining a healthy GI barrier. Gut microbiome imbalance throws off this balance and facilitates barrier weakening, right? The leaky gut. Again, allowing translocation of those PAMs and DAMPs. And these are increasingly being shown to harm the liver. It's this big loop, right? One aspect of it gets thrown off and it just keeps triggering down. It's like a domino effect. So this particular picture is really quite detailed. You may want to come back and spend a little bit of time with it. But this quote is something that I love. Complex interactions involve bacteria and intestinal barrier, which comes in close contact with dietary factors, food contaminants, drugs, chemicals, and bile. These elements can modulate the pathways linking the gut with the liver, leading to both local and systemic effects. The interplay is bidirectional. According to the available evidence, the majority of environmental factors leading to abnormalities in the gut-liver axis, and in turn to liver and metabolic dysfunction, could be managed. This is especially important in type 2 diabetes. So many of the underlying triggers can be modified. It's important for not just practitioners, but also patients to understand this. Remember, some of the feedback that they gave was that it was hopeless and that it was this life sentence. It isn't necessarily a life sentence, and it is most certainly not a death sentence. Okay. Brain, the gut-brain axis. Again, these pictures are awesome for people who are highly visual. This is a bidirectional communication between the gut and the brain, predominantly via the vagus nerve. Neurotransmitters like serotonin and dopamine can be directly produced by gut microbes, and their metabolism is also heavily influenced by microbes. So serotonin precursor tryptophan is driven away from the serotonin pathway and down the kynurenine pathway by bacteria in the gut. One of the ways that this happens is via LPS. And again, that's that bacterial produced endotoxin. And it stimulates tryptophan to go down the kynurenine pathway, which is away from serotonin. The kynurenine pathway is inflammatory and can lead to things like quinolinic acid. Quinolinic acid elevations have been implicated in type 2 diabetes, inflammation, and a number of hormone-mediated cancers. Short-chain fatty acids can directly stimulate the sympathetic, that fight or flight pathway, and stress causes an increase in cortisol, which can lead to increased GI permeability and increased inflammation. In BDNF, this is the brain-derived neurotropic factor. It binds to tyrosine kinase receptors. It plays a critical role in neuronal survival, synaptogenesis, and neuroplasticity. It's reduced in type 2 diabetes and increases with things like exercise. Interestingly enough, it's also been linked to impulsivity and risk-taking behavior. I love this picture. I love this quote. At a basic level, the gut microbiota interacts with the human host in a mutualistic relationship. The host intestine provides the bacteria with rich energy resources and an anaerobic, meaning lacking oxygen, protected environment in which to grow, and the bacterium aids in governing homeostasis within the host. It is mutualistic and bidirectional. So take care of your gut microbiome, and your gut microbiome will take care of you. We want happy little bugs. The gut immune axis. So we have the GALT, right? The gut-associated lymphoid tissue. We know that 70% of the human immune system resides in the gut. We have our mucus layer. This is the first line of defense in the GI. It's produced by goblet cells. It's a physical barrier to prevent host microbiota contact and translocation of things like LPS. The epithelial cells produce antimicrobial peptides. Those are those AMPs, MAMPs, PAMPs, DAMPs. And these are also things like antidefensins and Reg3 gamma. This is referred to as kind of this demilitarized zone. So IgA specific for commensals is produced with the help of dendritic cells. These are our antigen-presenting and phagocytic cells. They interact with B and T cells in the Peyer's patches, and they produce commensal-derived antigens based on microbiota sampling. Literally go out into the lumen of the intestine, grab something, bring it back, and say, "What's this?" Right? It's like the little kid that goes out to play. "Hey, mom, look. Hey, mom, look." And eventually the immune system's going to go, put it down, kill it, right? Short-chain fatty acids, these are just going to keep coming up. They're produced, again, by bacterial fermentation of indigestible carbohydrates called fiber, and butyrate, acetate, and propionate are our main examples. They actually create a hostile environment by lowering the pH. They have the ability to lower the virulence gene expression in microbes themselves, and especially butyrate, it regulates the size and the function of the regulatory T cell network. So another way to say this is that those short-chain fatty acids regulate the cells that act to suppress immune response. And these Tregs are critical in preventing autoimmunity. We have FOXP3 Treg cells. They suppress things like those NF-kappa beta cells we talked about earlier, and they maintain peripheral and mucosal homeostasis by sensing microbial products and metabolites. Production of bacteriocins. These are short toxic peptides produced by specific bacteria to inhibit colonization and growth of other species. It's one way that the microbiome actually polices itself and keeps the commensal balance within the gut lumen. Bile acids have antimicrobial properties as well. Those conjugated primary bile acids deconjugate to the secondary bile acids in the colon, and these are things like the deoxycholic and the lithocholic acids, and these can be highly toxic to certain bacteria, especially things like Staph A and C. diff. This is a good visual for balanced versus imbalance in the gut. And the quote points out what we just covered. An imbalanced microbiome creates an imbalanced gut, triggering immune reactions, not only in the gut, but potentially body-wide. We are going to talk briefly about the gut-skin axis, especially because of the issue with poor wound healing, slow wound healing, diabetic ulcers, etc. It's important to understand first that there is a gut-skin axis and how this comes into play. So vitamin D, studies show potential signaling pathway between microbiome and skin via vitamin D pathway. Probiotics can actually increase serum vitamin D levels in patients with a gut microbiome imbalance. And the large majority of skin issues occur in patients who also have gut dysbiosis. Hypothesis for this link is because of the LPS pathway, which tends to be upregulated in these particular pathways. Remember, LPS is this bacterial endotoxin. It can create a leaky barrier, really impeding, really skewing that permeability. Short chain fatty acid imbalance driven by imbalance in the gut microbiome and or a lack of fiber for the microbes to use. So microbes feed on fiber. And they make short chain fatty acids. So if the diet is healthy, if it's low in fiber, it's still going to be an issue. The microbiome induces IGF-1, so that insulin-like growth factor, and IGF-1 promotes proliferation of sebocytes and keratinocytes, as well as inducing lipid synthesis in the sebaceous glands. The epidermal function interactions are driven by these free phenol and paracresol. These are metabolites that are seen frequently after C. diff infections, and they can disrupt the epidermal barrier integrity by reducing the expression of keratin 10 and keratinocytes. Lowering these phenol levels has been shown to decrease skin sensitivity and improve the barrier function, improving wound healing. And the studies lower the phenol levels by giving probiotics orally. This is another quick pick to help you visualize and perhaps your patients visualize a healthy skin microbiome versus an unhealthy skin microbiome and the implications of those. I wanted to take a brief foray into aging. A large chunk of our type 2 diabetic population is considered elderly. If you remember the incident data slide, the first spike tends to be seen in the 45 to 55-year-old, and then there's a second spike in incidence seen in the 65 to 75-year-olds. It's a little bit of an aside. What age is defined as elderly varies and is very, very hotly debated. Currently, it stands at, in general, those over 85 are in the long-lived category, with centenarians in the extreme longevity category. By age 65, you win the label of late adult / elderly. So any test, any stool test that you look at least any stool test that you should be running, will show you a diversity score. And studies consistently show healthy gut microbiome has a good degree of diversity. And as the diversity decreases, the health of the gut microbiome and the health of the host decreases. We know microbial diversity decreases with age, as does the robustness of our immune system. Intestinal permeability also tends to increase with age. Inflammation or inflammaging is a term that has gained in popularity recently. We know that inflammation increases as we age. So the microbiome modulates immune response and inflammation. And is it any surprise that these go awry as the diversity of our microbiome decreases? Inflammation, it's like that one ring, it binds us all. So in inflammaging, we have this LGI, which is low-grade inflammation, and this increases as we age. Again, studies considering aging as 65 plus. The serum inflammatory markers that we tend to see elevated in this population are interleukin 6, interleukin 1, TNF-alpha, CRP, hsCRP, and fibrinogen. The microbiome modulates our inflammatory response from a very, very early age. There are some studies pointing to it now happening in utero. And again, it's not surprising that as the microbiome shifts away from healthy, we as a person do also. Let's pivot to the literature now, specifically looking at studies done on oral probiotic usage in type 2 diabetics and some of the most common comorbidities we see in this patient population. So the overall state of the literature, there have been a number of meta-studies studying probiotics for type 2 diabetics in randomized controlled trials on humans. In all of these studies, there was improvement consistently seen in hemoglobin A1C, blood pressure, CRP or hsCRP, fasting blood sugar, triglycerides, HDL, and HOMA-IR. Studies that use multi-species and multi-strain probiotics saw a larger improvement consistently across the board. Aspects that had inconsistent results, meaning findings in studies were conflicting. Some saw improvement and some saw no improvement. These were BMI, total cholesterol, LDL, and VLDL. The length of the treatment and whether they used a single species probiotic seemed to be largely related to these mixed results. So this is a study on patients with type 2 diabetes, hemoglobin over 7%, and neuropathy. They were not on any pharmaceuticals. They were given 15 billion CFU of a freeze-dried probiotic powder daily for 6 months. There was no other intervention, no dietary changes, no movement, nothing. On all of these slides looking at studies, you can see the specific formula that was used in this study in the little chart on the side. I will point out on this one, right, this is what you're seeing with one that has strains in it. So, it's that little letter number after the name of the bacteria. The results are really quite impressive on this monotherapy. There were drops in glucose, insulin, interleukin-6, CRP, and resistan. So resistan is this resistance to insulin. It's kind of how I remember it. It has been strongly linked to obesity, type 2 diabetes, inflammation, lipid imbalance, and a host of other issues. We also saw increases in adiponectin. So in this particular study, the researchers saw that the leptin decreased and the adiponectin increased. And so this is something we think about with this term called leptin resistance, right? Leptin is our long-term energy and storage. It's our satiety hormone. It should be low when we are hungry or we have low fat stores, and it should be high when we are well-fed and have high fat stores. In obesity, there's a lot of leptin. It's made by adipocytes, and the brain stops listening. It just shuts down. Adiponectin increases with insulin sensitivity, and it's a highly anti-inflammatory. And that ratio improved in this study. As a side note, I will say that adiponectin can have they're truly low in patients who have a very low body fat percentage. Adiponectin is also made by adipocytes. If you have low body fat, you can't make it. There were no adverse events in this particular study. This next study is looking at a probiotic targeting endotoxin in type 2 diabetics. It was a randomized controlled trial on patients who had already been diagnosed with type 2 diabetes. They got 2 grams of freeze-dried multi-species powder every day for 12 weeks. And there were, again, no additional interventions. There were improvements in waist to hip ratio, blood pressure, mean arterial pressure, and HOMA-IR. For those of you who aren't familiar, HOMA-IR is a measure of insulin resistance that's based on fasting glucose and fasting insulin. There were also improvements in C-peptide. This is a substance made in the pancreas along with insulin. In this particular study's triglycerides, LDL-C, and endotoxin all improved. And so endotoxin is a substance that is thought to induce inflammation and obesity, specifically in metabolic syndrome and type 2 diabetes. And again, there were no adverse events. So I'm going to point out for a moment, no adverse events in a study is a really big deal. When looking at studies and looking at, am I going to use whatever this particular study used to achieve these results for this one patient, sometimes those adverse events can help you determine one intervention over another. Were there adverse events? Yes or no? And was there an adverse event that you think your patient could handle, that it won't be that big of a deal? Or do you know that if that adverse event happens, you've lost that patient, they're gone? This particular study is looking at obesity and depression and psychobiotic usage. Remember, depression was one of the top 4 comorbidities in type 2 diabetes. This study was done in normal weight, obese, and pre-obese and obese women. They were given a powder containing 13.5 billion CFU for 3 weeks and no other interventions. As a little bit of an aside, a normal weight obese person, they have a normal BMI, but a very high body fat percent. They also tend to have high oxidative stress, inflammatory markers, and some metabolic markers. So for example, random elevated fasting glucoses or kind of borderline hypertension. They're at higher risk for cardiovascular and metabolic diseases. The results that we're seeing here were a decrease in BMI, a waist to hip ratio, the total body fat percent went down, and the free fat mass went down. This particular study also saw improvements in body image and body uneasiness, and this was evaluated via the Eating Disorder Inventory Questionnaire. And it's very widely used self-evaluation for disordered eating. It looks at things like drive for thinness, body dissatisfaction, perfectionism, maturity fears, impulse regulation, social insecurity, et cetera, et cetera. And again, there were no adverse events here. This particular study focused on depression specifically. And in this study, patients were given a multi-strain freeze-dried powder probiotic in a 7.5 billion CFU dosage. I really like this study because they looked at both objective, which are the fMRI studies, as well as subjective, which are the PANA studies. The fMRI, and these are MRIs done while looking at pictures, answering questions, etc. With the probiotic, what was seen was an increase in functional connectivity in the salience network. This particular network is involved in things like attention, perception, and cognition, and a decrease functionality and functional connectivity in the default mode network. This particular network is responsible for things like distraction, rumination, impaired decision-making, and impaired reasoning. Questionnaires are attempting to objectively evaluate something which is inherently subjective, right? So objectively trying to evaluate our thoughts and our moods and our feelings. And the PANAS is very commonly used. It evaluates positive and negative affect. And it lists emotions like I am distressed, interested, proud, irritable, nervous, etc., and then rates the frequency of the emotion felt over the past week from slightly or not at all the way through to extremely. And then the scores are based on just the positive emotions and then again, just on the negative emotions. In this particular study, we saw that you can see here that the positive affects, so those positive emotions, increase substantially. And the negative ones, the things that are hopelessness and risk aversion, those went down. I'm a mom of 2 young boys, and decreased risk aversion scares the crap out of me. However, in the large majority of adults, risk aversion looks like being afraid to say hello to a coworker, or being afraid to leave your home, or thinking that it's too risky because everybody probably hates you, right? It's that sitting at home by yourself and ruminating type risk aversion. And so we do want that to go down. There were a surprising number of studies done on oral probiotics and wound healing. And the results pretty consistently showed improvements in the length, depth, and width of the ulcer, the need for skin graft and body surface area grafted, the need for frequent dressing changes went down, quality of life went up, fasting glucose went down, insulin improvements, hemoglobin A1C improvements, total cholesterol and CRP improvements. And that's with oral probiotics. So for this particular study, I pulled it for this talk because One of the things assessed was bacteria in inguinal skin folds, which is a big deal, especially in our type 2 diabetics that have had the disease for quite a while and in people who are struggling with obesity. So the multi-strain freeze-dried powder probiotics were given to patients who had been colonized with multi-drug resistant gram-negative bacteria. The levels of this MGRN were monitored with both stool samples and inguinal fold swabs. The number of patients with MGRN was down 42% by 8 weeks into the treatment protocol, which is pretty stunning. And at 24 weeks, so this is 12 weeks after they stopped the probiotic, that result was still seen. So it was still seen that 42% of patients did not have the MGRN. And they didn't see an uptick in that multi-drug resistant gram-negative bacteria until week 36, which was a full 24 weeks after the treatment had stopped. And no other interventions were done. This particular study was assessing a pre and a probiotic targeting inflammation and oxidative stress in type 2 diabetics. It was a randomized controlled trial on patients already diagnosed. They were given freeze-dried multi-species 21 billion CFU probiotic powder. It also had 100 milligrams of fructooligosaccharides in it, or FOS, and they were given this therapy for 8 weeks. They had no additional interventions. We saw improvements in inflammatory markers, as well as improvement in serum glutathione levels, suggesting a reduction in oxidative stress, right? You have a lower need for depleting your glutathione. And again, there were no adverse events. This is another study looking at inflammation and oxidative stress in type 2 diabetics. I really like it because it looked at inflammatory markers in addition to other markers, and they were given this particular powder for 6 weeks. Again, no additional interventions. And the malondialdehyde, which is the MDA, it's one of the final products of PUFA peroxidation in the cells, and an increase in free radicals causes an overproduction of MDA. The level is commonly known as a marker of oxidative stress and antioxidant status. And then we saw that there were, in addition to the decrease of MDA, there were decreases in interleukin-6, triglycerides, insulin, HOMA-IR, etc. And again, no adverse events. So now that we've spent a little time looking at clinical studies showing us how impactful oral probiotic therapy can be to type 2 diabetic patients, not just for the positive impacts on traditional markers like glucose and insulin, but also on lipids, inflammatory markers, and oxidative stress markers. Not to mention the positive impact they can have on a number of the common type 2 diabetic comorbidities like depression. Let's spend just a few minutes reviewing how to pick a quality probiotic. There are a ton out there, and it is very important to make an educated decision and be able to explain to your patients why you are recommending they take one product over another product, especially when it is becoming increasingly easy for patients to get their hands on products that may or may not be the quality you would like them to have. This is a slide that perhaps should have been at the very beginning in the definitions. It's what's a probiotic, but I kept it here so that everything kind of flows together. A probiotic is a live microorganism that, when administered in adequate amounts, confer a health benefit on the host. You'll notice this graphic starts with strain up at the top, and a strain that has been characterized. I'll take a look at that in a sec. It is safe for consumption. And there is a focus on human clinical trials showing efficacy at the dosage that's being recommended. And then they also evaluate things like shelf life stability and, again, coming back to shelf life stability on the dosage that was seen to be beneficial in those human trials. So strains. The characterization word is really just its ID based on biological, physiological, molecular, etc. And 16S rRNA is PCR, in case that's still a question for anybody. So both 16S and whole genome shotgun sequencing are currently used. Characterization tells us things like the environment that the microbe will survive in. Will it live in high pH? What about low pH? What about all of the other gastric secretions? It also tells us the actions. Does this particular organism modulate inflammation or immune cells? What about lipids? Does it have the ability to either directly kill or inhibit colonization of other microbes? And what products does this particular microbe create? I put this slide in here specifically because so many of the meta studies done on oral probiotics in type 2 diabetics pointed out that they saw consistent positive results and greater positive results when a multi-species, multi-strain probiotics was used. So how the strains that make up a probiotic come together is important. You can't just put probiotics that have been shown to positively impact a specific symptom in a bottle and call it good. Some will compete with each other and actually kill or inhibit the actions of the others in the bottle. Some combinations have additive or complementary impacts. Studies looking at single strain supplementation versus multi-strain supplementation have consistently shown greater benefit with multi-strains. The hypothesis behind this is this concept of quorum sensing, which is the cell-to-cell communication. And it's based on the production, secretion, and detection of small signaling molecules given off by microbes. This can be helpful, so they can be additive and very synergistic, or it can be harmful and it can trigger those bacteriocins that we talked about earlier that would then inhibit or kill other microbes in the mix. This particular study on this slide has some really cool examples of both of these things happening if you want to spend time geeking out on something. So what this means is that the actual product in the bottle needs to be tested to ensure it achieves what the product claims to achieve. Again, just throwing strains together is not adequate. You cannot guarantee outcomes without testing the actual formulation in the bottle to see if it does what you say the individual things do. So this is a summary slide. It's kind of a little cheat sheet checklist for picking a good probiotic. Here are the basics. Multi-species, multi-strain product. The product should have the strains listed, right? It should have something after the genus and the species. This is what gets you into those very, very, very targeted and specific action. There should be some indication of third quality certification, like a CGMP or something like that. As with all things in our world currently, there are certifying bodies, and those exist in the supplement world as well. This actually helps the consumers, practitioners, patients, wholesalers know that at least some level of compliance and quality control has been achieved. You, as the practitioner, should also do a little bit of lit search here. Do not rely 100% on the company. Be a savvy consumer and advocate for your patients and your clients. Pop into the literature and see what is being said about those particular organisms. The probiotics also need to arrive and survive their journey to the shelf through the GI and still have metabolic activity. The manufacturer should have studies showing that this happens. The entire probiotic formulation should have studies showing efficacy, preferably in humans, and the organisms in the probiotic will interact with each other, not just the host, and sometimes they don't play well. So having the studies on the specific formulation helps you as a practitioner know that the outcome you're wanting is the outcome that you have a strong likelihood of getting. Any company that you are purchasing from, probiotics or otherwise, should be able to point you to the studies that were done on their products. They should be on their website. If they're not, pick up the phone, send them an email, and ask them for their studies. Companies spend a lot of money on them, and they really and truly want you to ask for them. The more that our industry demands this type of information, the more the companies who are hesitant to spend the money on it will do it. And in turn, the companies who spend the money on it, their quality and efficacy of products just goes up and up and up and up. Everybody wins. So let's take a moment to look at some of these key high-level takeaways. The gut microbiome and its many, many axes have links to all of the systems that are impacted by type 2 diabetes. Hopefully, you're screening your patients with type 2 diabetes for comorbidities, including depression. And as you've seen, targeted manipulation of the gut microbiome can have positive impacts, not just on type 2 diabetes, but on the many associated comorbidities. And as Hippocrates said 2,500 years ago, it really does all start in the gut. Thank you so much for your time and attention, and I hope you enjoy the rest of the conference.